Hello guys. So we are going to start on the second session of these lecture series for the first exam. There was something in epithelial tissue I forgot to mention. See, when I sleep at nights, I think about you guys and I think about what I said during the lecture. So there is something I forgot to mention. Should I go back and mention it? Yeah, I should go back and mention it right here. Okay, so I think they did say it in here. Maybe I don't know. Let me move this thing around. They did not. Transitional. No, I said come back to that one. Stratify. Oh, yeah, I think they mentioned it here. I overlooked it. Okay. Look, on the bottom of the this tissue, this gland, whatever it is, so you have like cuboidal epithelial tissue on the bottom. You have cuboidal, and on the top of this gland or whatever structure you have, again, there's squamous epithelial tissue. So what do you call that? Whatever is on top, and then the bottom is basement membrane. Right here is a basement, they call it basement membrane, and then these are possibly connective tissue or smooth muscle, what have you. Then that top, this is a real picture right here. So the top is squamous epithelial tissue, on the bottom is cuboidal epithelial tissue. So whatever is on top, you call this, of course, stratified squamous epithelial tissue. I hope that I forgot to mention. So whatever is on top, away from basement membrane, all of the epithelial tissues, all of them are attached to a basement membrane, and that's called, which is a connective tissue. Okay. So, and then underneath it could be smooth muscle. In this case, it's smooth muscle or whatever, another layer of connective tissue. So on top, these are all squamous epithelial tissue on top right here. So this tissue is stratified squamous epithelial tissue. I did not mention that last time. Sorry about that. Oh, I jumped too much. Okay, no, I did not jump too much. Okay, so let's move on. And I talked about system and everything else. I talked about nerves and how neurons, and the last time video is in YouTube under tissue and what did I call it? Tissue and nerves. So Bio 2, tissue and nerves. And I'll put this one on whatever comes up, supports and protection and movement. So this is your integumentary system. So we talked about, I gave you a rundown of it. When you take anatomy, there are many more layers, but for this class, your textbook mentioned 3 layers, and we stuck with these 3 layers. The epidermis right here, dermis underneath of epidermis, and hypodermis or subcutaneous, another name for it. It is the third more less here. This, of course, is outside of your body, and then from there and on. So the epidermis is made up of cornified, this layer is called cornified, stratified squamous epithelial tissue. And then underneath of that, right here, dermis is made up of dense, irregular connective tissue. And then underneath of that, as soon as you see fat cells, adipocytes, these are fat cells, adipocytes, as soon as you see those, that's subcutaneous or hypodermis. Okay. In the dermis layer, you have, of course, the hair follicle attached to this erector pili that causes the hair to become goosebumps. You only get goosebumps when you're cold and when you're in fear. Okay, so and then there are other nerves which I'm not worried about it. You don't worry about it. You cannot see them on microscopic slides. I think I forget some stuff to mention in here. It will come up. Here's the microscopic slides of it. Right here is the top. You can see from here to here. All of that is your keratinized, stratified, squamous epithelial tissue. all of this right here, from here to here, all the way, it's your dermis layer, which is made up of a dense, irregular connective tissue. And right here is adipocytes. That adipocytes right here is made up of fat cells, adipocytes. And that's what layer that is. That's a subcutaneous or hypodermis. So let's see. Integumentary system, hair, setae, you don't know what setae is, do not worry about it, but you know what scales are, feathers, they're all are part of the integumentary system. Of course, in human, the integumentary system is not as complicated as some other species like birds or fishes and other organisms. And then, of course, integumentary system is for protection. And conified, stratified squamous epithelial tissue, I already talked about that. It comes from ectoderm. You don't know what ectoderm is. Do not worry about it. I will not ask anything about it. But for next exam, then definitely you should know what that sentence means. And dermis is a true skin, which comes from mesoderm, which we'll talk about it later. What is mesoderm? Okay, that term right here. Okay, oops, sorry. Anyhow, dermis contains connective tissue, blood vessels, pigment cells, fat cells, keratinization. Okay, let me go over, let me go over keratinization here in a minute, next slide. And then subcutaneous layer is the most, the hypodermis, or subcutaneous is the layer on the bottom. So what is keratinization? So I'm answering, this is the model we have in the lab. So the very bottom layer, these cells undergo mitosis right here. They undergo mitosis, and of course, these are your squamous epithelial tissue. And then they push up these cells to that layer. As soon as they get here, the cells will die, the nucleus will die. And the only thing that is remain from these cells, the remaining are keratin. Keratin is a protein molecule, which are right here. They accumulate, and they do cover up the sorry about that. Let me turn this down. Okay, they cover up the stratified squamous epithelial tissue, and that's why this tissue during exam or any other time, you should say it's cornified or keratinized or horny stratified squamous epithelial tissue. So when the cells get up here, they die, and what is left of them is keratin. Keratin is a protein molecule. Another protein molecule here you should know is collagen. That's the most abundant protein molecule on planet Earth, collagen. So all of this dense irregular connective tissue in this area, not the blood vessels, not the gland, all of these area, the creamish area, not these blood vessels, not these nerves, none of those. All of these, this is the smooth muscle, erector pili, hair follicle, all of that stuff right here is made up of collagen and dense irregular connective tissue. So in the integumentary system, you should know two protein molecules, that one is here is keratin and one down here is collagen. Okay, so I think I talked about everything. Chromatophores, when we talk about other animals a little bit, you should know a few things about other animals too in this section. And one of the things, well, melanin, we humans have melanin that causes our skin to become darkened when we stay in the sun, become black brown. Keratinoid is another pigment in our skin, which is orangish color. Xanthophore is yellow color in our pigments. And iridophore, we do not have it. We humans do not have it. And these are contains crystals of guanine. You remember from your Bio 1, guanine, or other purines. The purines are, you remember that they are two rings of the nitrogenous bases. You remember that, two rings. And then pyrimidines are one ring of nitrogenous bases. And then, so these are crystals in animals like octopus, cuttlefish, that they can change the color based on the environment they're surrounding in. So we humans do not have that iridophores, but these animals like, as I said, octopus, some squids, some frogs, they can change their color because of this, the iridophore. Let's talk about muscles. Again, I promised myself I will talk only for 1 hour, so you guys will not fall asleep. Skeletal muscle, they provide support. Exoskeleton, of course, we do not have it. It's found in arthropods, which we'll cover it up later on during semester. By late April, we'll talk about arthropods. Endoskeleton, that's what we have. Arthropods do not have endoskeleton. We have endoskeletons. And our endoskeleton is made up of calcium, phosphorus, and protein molecule. Okay. Chondrocytes, they are cartilage cells. That's chondro, it means cartilage. Cyte, it means cells. And hyaline cartilage is a clear and glossy. And different types of bone development is endo, I should mention maybe, and osteocytes are the bone cells, okay? So anyhow, different types of bone development, it means when you were born, what happens to you? There are, they call it ossification, bone development, when the bone becomes harder. Endochondral ossification, it means at the end of your long bones, there is a ring of cartilage. And as you're growing taller, that ring of cartilage becomes bone. It's not cartilage anymore. But that, there are different theories. People say that puberty stops. Some others say at age 25, you take a grab for grab, but most people agree that at age of puberty, you stop growing your height, okay? But other people say, no, it can go on until, it's not much after puberty, it's not much, but it can go all the way up to age 25. But anyhow, that's endochondral ossification. The other one is intramembranous ossification, and that is when you're born between these plates of your the bones in your head. They call them sutures. They are soft tissues here, and then those soft tissues, membranes, actually, those soft tissues membrane, they become hardened by age of 1, 2, okay, give and take, somewhere in that neighborhood. And that is called intramembranous ossifications. And they are found in the skull, as I said, and your clavicle, your shoulder bone. Yes, they can be found in there. They're just membrane, that's what they call them, intramembranous. Okay, so let's talk about the histology of your bone. As you know, the long bones we have in our bodies, it is right here, it is surrounded by compact bone, and in the middle, you have the bone marrow. Right here is the bone marrow where the blood vessels, fat, lymph, all of that are. But right here, from here to here, from here to here, that is your compact bone, and that's what I want to focus on talking about right here. That's compact bone. So right here, when compact bone is going to become bone marrow, as I talked about it, right here, it becoming bone marrow, there is an area, a small area that is spongy bone. So compact bone in the around of your, if this is your bone, and then so all around is compact bone, and then you go inward, there's spongy bone, and then it's bone marrow right in the middle. I hope I'm making some sense. So I'm looking at the compact bone, which is that's a histology of it, and this area, all of this you see, that is your spongy bone. Okay, so this is all compact bone. I don't want to draw too many. This is all your compact bone. Okay, so in the compact bone, you do have osteon This is an osteon. So in here you see 1, 2, 3, 4, 5, 6, 7 osteon, for example. And they just cut a piece of the compact bone and showing it to you. So the osteons go all around, and then spongy bone, and then the medulla, your bone marrow, sorry about that. So the structure of the osteon I do have right here. This is an osteon right here. They're showing you osteon. And then in this model in the lab, which you have, this is then osteon, this is osteon, 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 osteon. So let's go ahead and talk about osteon. I might come back and forth right here. Is an osteon, right in the middle of it, you have, they call this central canal. In the central canal, you have arterioles, venules, and nerve, okay? And it is surrounded by osteocytes. These are all your osteocytes. And between the osteocytes, this area is calcium and phosphorus, compact bone, hard bone. The osteocytes are connected together by that structure, little line like a spider legs. Okay, these are the spiders, and these are the legs of the spiders. They're all connected together by a little canal. Those little canals are called canaliculi. Okay, so right here is your, if you would, it is your matrix of the bone. They call it matrix of the bone. And then right here is the osteocytes connected. So food comes from these blood vessels. They sip through the canaliculi. They get to the osteocytes. From that osteocytes, they sip through canaliculi, and they go to that osteocytes. And from that osteocytes, sips in the osteocytes, and so on and so forth. Okay, so food and nutrients, and of course, oxygen and carbon dioxide and waste material from the bone go back that way into the blood vessels, into the venules, and the venules take it, which we will talk about it later on. So this is a picture of an osteon. And what we have right here, these are all osteon. And here is showing that an osteon is connected together, central canal connected together by perforating canal. And these are osteocytes, 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 osteocytes. And these little lines you see around the osteocytes are canaliculi. And this whitish area, that is your matrix, which is made up of compact bone. So here I think everything is mentioned that I talked about. I hope it's showing your osteon, central canal. Periosteum is a connective tissue surrounding the whole entire bone. And then these are perforating fibers, which the blood vessels from outside of the body. They did not show it in here. No, they did not show it. They should have shown a blood vessels, one blood vessel come to the bone like that. Okay, so the blood goes into, and you can see the nice, beautiful canaliculi right here, and these are osteocyte, 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 and in between the canaliculi are your compact bone, which is, well, the matrix right here. So let's go back to this. This is matrix right here. These little hair-looking things, that's canaliculi. This is osteocyte, 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 osteocyte. Okay, so if you would, these central canals are connected together by perforating canal, and that is a perforating canal. This is central canal, central canal, central canal. So this is a central canal connecting to another central canal. That central canal is connecting to another central canal. And these canals right here is called perforating canal, and that is called central canal. So central canals run this way in my bones. Perforating canals run this way in my bones. Okay. Right here, they are showing you a longitudinal cut of the bone, and right here are central canal, perforating canal, perforating canal. I hope I'm making some sense. And of course, this is spongy bone. All of this is spongy bone, and this is compact bone, of course. Okay. So let's go. I think I talked about these things too much. And it's showing you the canaliculi. Oh, lacuna, you have, you cannot see it. What we have, okay, on these, well, on the cross section, you will see it in the lab. These cells on the sides, if you would, you will see an orange marking outside of it. That is lacuna, the empty space outside of the osteocytes. And osteocytes in this model, they are blue dots. Okay, when you go to the lab and looking at the lab material, you will see that. Okay, so canaliculi, I talked about that. Concentric lamellae. Okay, lamellae, do not worry about it. Lamellae is one ring. It's called lamellae. Another ring is called lamellae. Okay. So matrix is made up of calcium phosphorus, lacuna, canaliculi, osteocytes. I talked about everything. Okay, I talked about histology of bone, you guys. And then these are the name of the bones. I would like you to know them for the lab practical exam. You probably had anatomy, you know them already, great. If you don't, please learn them. We do need to know some of these for later on during semester, okay? When we talk about arthropods and other animals, we need to know them. So axial bones, by definition, it includes the skulls, all of the bones that are in x-axis, that's what they call them, axial bones. They are in x-axis. So the skull would be one, vertebral column, sternum, right here, your bone right here is called sternum, and the ribs. Okay, so these are all the bones that are in x-axis and connecting to the bones. Appendicular bones are bones that are limbs and pectoral and pelvic girdles. All of those are referred to as appendicular bones. So I would like you to know them for purposes of the lab practical and everything else. Vertebral column, so you should know the very first bone is atlas and the second bone is axis. So I wrote them down as the first cervical. Cervical, it means the bones in the neck area. So there are 6 or 7 of them in here. And then it supports the skull while allowing it to pivot. Axis is the second cervical bone. I have to turn axis. Okay, I hope I'm making some sense. That's why I think it's important. And then the second set of your vertebral column are there are 12 of them. They are your thoracic bones, okay, in back. And lower back are lumbar. So I would like you to be familiar with these terms: cervical, thoracic, lumbar. And then you have sacrum. There are 5 bones that are connected together, and coccyx toward the end of your body, if I may. So coccyx, again, these are your tailbone, which the tailbone right here, if you were born with a longer tailbone than normal, right after you were born, the surgeons will cut it off. Do not worry about any more. That's all I want you to know in anatomy. You spend a lot of times dealing with these bones, but don't worry about it for my purposes in here. Cancellous bone is spongy bone. Another name for spongy bone is called cancellous bone. And compact bone, we talked about it. Osteoclast, they are bone reabsorbing. It means they destroy bone. And then osteoblast, they build bone. And I will talk about them hopefully soon, or calcium right now, yeah. Calcitonin, lower calcium level in the blood, and parathyroid hormone increase the level of calcium in the blood. And so what happens, so let's talk about it. What happens, your bones, the bones of your body constantly are being remodeled. This is not the same bone you had 5, 6, 7 years ago. So what happens, the hormone calcitonin, it comes from your thyroid gland, and in the back of your thyroid gland, your thyroid does not go in all the way back. It's a thyroid gland. Your thyroid glands are like a butterfly, and in the back of it, there are 4 parathyroid glands. These parathyroid glands release the hormone parathyroid, the calcium level in the blood by breaking down the bone. So the level of calcium in your blood fluctuates low and a high, low and a high, low and a high, low and a high. Of course, your diet is involved as well, and your kidney. I'm giving you just a small snapshot of what's going on. Okay, so calcitonin from your thyroid gland goes to the bone and break down the bone. So calcium level in your, I'm sorry, the other way around. Calcitonin from your thyroid gland, it goes to your, it lowers the calcium level. So in your blood, so it takes the calcium from your blood and give it to the cells of osteoblast, and osteoblasts start laying down bone. They make bone. Okay, so calcitonin take the blood away from your blood vessels. Here's the blood vessels and calcium in it. It takes away blood. It takes away calcium from blood, and it takes it to the bone. That's calcitonin and osteoblasts at work together. These two work together. And then these two work together. Parathyroid hormone is released and it breaks down the bone. So calcium goes back. You need calcium in your blood. Calcium goes back by help of that hormone. Okay. And then the bones that are breaking down, they're being reabsorbed. That's the word your textbook uses. They have to be built again. Okay. Anyhow, muscular movement, sliding filament is okay. Only talk for half an hour, you guys. Sliding filament model. That's what I'm going to talk about. Atrophy, it means if you don't use your muscles, it doesn't happen in your case. But people who are paralyzed, that's why they go and move them and shake them and so on and so forth. Because atrophy does not happen, the muscle cells do not die, and that's when atrophy means. Atrophy means muscle cells are dying. Hypertrophy means the size of muscle cells increase. From the time you were born until the time you die, the number of your muscle cells in your body is constant, and also the number of the neurons. Okay, they do not change. They're not supposed to change. So when you go to the gym and you pump iron, what happens? The size of your cells, muscle cells, increase, and that is called hypertrophy. If in case your muscle cells number increase, the numbers increase, God forbid, that's an abnormality. Houston, we've got a problem. I don't know, you guys watched Apollo 17. Oh, 13, not 17. Apollo 13, you watch it. Houston, you've got a problem. That's some kind of cancer sarcoma. So the numbers cannot increase of the muscle cells in your body. It's just the size supposed to increase. Okay. And then, for example, pregnant women, when they become pregnant, right, the uterus, the numbers do not increase. The size of each cell increases, not the numbers. Okay. Anyhow, give you. When you go to the gym, I give you. Okay, another name for sliding filament model is attack pull release cycle, which is 50 to 100 times per second. And then creatine phosphate, when you pump iron, when you run fast, you run out of ATP, but there is a reservoir in your muscle cells called creatine phosphate. I'll go over all of that later on in detail. So there is a reservoir called creatine phosphate that it has phosphate attached to it. It gives its phosphate to ADP, and it puts out ATP so you can run 20 miles. Okay. And then red muscles, I'm not too crazy about this. Red muscle fibers, they're highly vascularized and have many mitochondria. And white muscle fibers, your fast fibers, fast twitch, like in your eyes, in your muscles, all your body, your biceps and your quads, you have combination of both. Okay, but for example, here in your biceps, you have less of white and you have more of red. In your eyes, right here, you have more of white than red. Okay. Here are the 3 different types of muscle. We showed it to you at the beginning of semester. I'm not going to spend time on it. Okay, what happens? So, for example, this is a muscle cell, skeletal muscle cell. As I said, the nucleus is on the periphery. Let me put my sarcolemma is the cell membrane of the muscle cells. And then sarcoplasmic reticulum, they are smooth ER. My handwriting is improving. Huh? Smooth endoplasmic reticulum in Bio 1, you talked about it, is full of calcium right now, this one. And then you have A band, I band, and myofibril. So, and sarcomere, Z line. So I should talk about all of those muscle cells and nucleus and fascicles. So anyhow, so your muscle bundle is made up of fascicles. Inside of fascicles, you have the muscle fibers, and then the muscle cells are drawn in here. So A band, and I will talk about this in some detail later on, but just right now giving you a prelude and an introduction to what is coming at you. So A band is combination of thick filament and thin filament. Just write it down somewhere, we'll come back to it. I band is just thin filament by definition. I'm giving you just a definition. Z lines, that's where I band attaches, the thin filament attaches. Thin filament or actin, you might see that somewhere. Thick filament or myosin, okay? So what is the definition of sarcomere? Sarcomere is unit of a muscle cell. One unit of muscle cells is called sarcomere. And from Z line to Z line, right here to here is called one sarcomere. And you will see it in a minute. And then you have H zone, which I will come to it later on. Let's go. Here they are, the nucleus are in the periphery, and here are the band. You see the striation. That striation is your myosin. Okay, great. So the myosin is made up of many. Here is a thick filament. Right here is called thick filament. That thick filament is made up of these myosin molecules, which have a head and a tail right here, okay? And there are many of them in the thick filament. The thin filament, these are thin filament. The thin filament is just not one protein molecule. It is made up of these actin molecules, if you would, tennis balls. And then it's by a rope is connected together, is called tropomyosin. So actin, and that's a rope right here. Have you seen those balloons that are connected together? So, and then there are little ping pong balls right here. Those ping pong balls are called troponin complex right here. Those ping pong balls called troponin complex. And that is the structure of the thin filament or actin filament. Actin filament, or another name for it is called thin filament. Myosin filament or thick filament. Okay, I hope I'm making some sense, everybody. Let's move on. Here they are. They're showing you 2 polypeptides coil around in a supercoil myosin molecule, and then there are many of them in the thick filament. Okay, same thing again. I'll show them to you in detail. Now, what happens When the muscle is relaxed, this is when the muscle is relaxed. You look at this is the Z line, right? And from Z line to Z line by definition is called sarcomere, right? This is your thin filament. Right here is thin filament. Here is a thick filament. Here, come on, I have to write down the whole thing. Thin filament, this one, okay? And then when muscle contract, look what happened. And this area, which there is only myosin, right, is called H zone, okay? There is no actin, there is nothing in there, just myosin. Remember, A band is from when there is both actin and myosin, right here. Oops, sorry, is A band. And then from here to the next one is I band. Remember that? So, and now look at it when the muscle is contracted. Look, you see the difference? Z lines are closer together. H zone is shorter, or in some cases disappear. Nothing happens to the A band, and I band, of course, they get smaller. Let's look at it. Same thing again, relaxed, and compare them. Compare the two together, and you will see the difference. You should be able to see the difference. And then, oh, okay. So I didn't talk about, well, I'll come back and visit that. Hang on. Just like movies, I like to go on and keep the suspense and then come back to it, you know, start a different story. And then they, at the end, they come back and put the two stories together. Hang on. So what happens, you remember, these are your neurons, your motor neurons. You remember that at the end of the neuron, there are axon knob. The axon knob release neurotransmitter. Now the postsynaptic neuron is not a neuron, it's a muscle cell. So what happens when these are ACH, remember, acetylcholine, are released and attached to these receptors, and what happens, sodium gets into the muscle cells. When sodium gets into the muscle cells, then it causes calcium to be released from sarcoplasmic reticulum. Okay, when calcium is released from sarcoplasmic reticulum because of the neuron, if the neuron is cut off, the muscles cannot contract. You know that, people who became paralyzed. Neuron cannot release neurotransmitter, has been cut off, muscle cannot contract. You know that, okay? So what happens when the calcium is released from sarcoplasmic reticulum right here, I'm talking about right here, calcium is released from sarcoplasmic reticulum, those calciums comes and attach to the ping pong balls right here, which is called troponin molecules, right? So the calcium attaches to troponin, and when calcium attaches to troponin complex, what happens? The shape of the actin changes. If the actin is like this and during relax, it changes and becomes like this. When it changes its shape, become like this, the head of the myosin attached to them. When it's like this, the head of the myosin are not attaching to them. So when the head of myosin attached to them, then attack, pull, release. They pull them, release, attach again, release, attach again, release, and that's why the Z lines, they come together. That's why when a muscle is contracted, it becomes shorter. When it's relaxed, it's longer. Okay, the muscle. So the head of the myosin, as I said, attaches to these actin molecules right here, to these actin molecules. The head of the myosin attaches to the actin, okay? The only thing troponin complex is the calcium attaches to it. And then when they release, when they are releasing, you need ATP for it. Right here is attached, and when detach, ATP, as you know from biology 1 becomes ADP plus PI, right? That is one use of ATP you learn in biology 1 that in muscle contractions. Right now I'm using my hand, I'm talking, I'm using a lot of ATP in my body, okay? So for detachment. And that's why when people die, this detachment does not occur, it stays attached, the muscles stay attached, and that's called rigor mortis. Rigor means stuff. And this is a nice diagram, guys. Please, you go over it and study it in detail. If you have any question, let me know. I'm going to go over it. If you have any question, it puts everything, it's not in your textbook, it puts everything together that I already mentioned. Here is a neuron, release acetylcholine, right? And then the acetylcholine here causes sodium to be released into the muscle cells. So now inside of muscle cells, you have a lot of sodium. That sodium, you have action potential in muscle cells. That sodium in muscle cells causes a calcium to be released. All of these calciums are released and they go sit on the thin filament right here of the troponin. Then the head of the myosin attach. Right? The head of the myosin attach, and of course, bring them together, release, bring them together, release, bring them together, release. And then what happens? The muscle contract. At the end, all of these calcium must go back into sarcoplasmic reticulum, which ATP is needed again. So you need two places you need ATP. One, for the calcium to be pumped back into the sarcoplasmic reticulum. And the second one, each head of the actin, each head of the myosin need ATP to be released from actin molecule. Detach. Okay, so read that. And then sliding filament model, I talked about it. Atrophy, I already mentioned that. Hypertrophy, hyperplasia, I'm just checking myself. See, attack pull release, how many of them happens per second? 50 to 100. If you were wondering about that one, I never asked that number. They call them cross bridges. When the head of the myosin attaches to the actin molecule, it's called cross bridges. And then pull it, release, pull it, ATP needed to release, pull it, release, pull it, release until you cannot do it. And as a result of this, creatine phosphate, you run out of ATP. Creatine, athletes are taking creatine now, they drink it as a shake, as a whatever it is, as a drink, and then the amount of creatine goes up. So the phosphate attached to them so they can compete faster, longer, whatever. Red muscles, white muscles. I forgot something. Okay, I guess I might as well. When a muscle is contracted, I should write it down because some of you guys complain later on and say, oh, what is this? I didn't get that. So when a muscle is contracted, A band gets shorter, right? And remember by the definition of A band is from here to here. Oh, sorry. I should. Sorry. A band, well, it's very close to each other. So let me go back. This is better. A band is from here to here. So you can see I did not, that's A band. I band, this diagram doesn't show it, is from here to next myosin. So that would be I band, okay? And then of course, this is H zone. So Z line, Z line. So when the muscle contract, I band gets shorter, A band gets shorter, I band, come on, I band, no, I band stays same, A band stay the same, I band gets shorter, and H zone almost disappear or shorter. And then Z lines get closer to each other. Sarcomere, what happens to that? What happens to sarcomere? Get shorter, right? And then all of that, you guys, is up right here. Remember, A band is from actin and myosin. I band is just actin, of course, plus Z lines is in between. So A band, nothing happens to it, stays the same. Shorter, I write it here that you know that. Z lines closer to each other. Sarcomere, closer, shorter. Okay. And then H zone is not here. H zone almost disappear or it gets shorter. Depends on the muscle. Okay. I think I talked about everything. I like to stop at homeostasis, and then I don't want to get you guys bored with everything that I have to say. And if you have any question, please let me know.